Today I'm going to be talking about feline immunodeficiency virus, other known as FIV, and my name is Zoe Stevens. FIV was first noticed in the 1980s when a cat colony was found to have a high prevalence of opportunistic infections and degenerative conditions. FIV is closely related to HIV, which is the human immunodeficiency virus, and was very devastating for the human population when it first came into the genome. They are both types of lentiviruses, and FIV has 5 subtypes based on the nucleotide sequence differing in the coding for the viral envelope. Among the different clades, there is also variation. Cross-species is uncommon, but there is a case found in bobcats and pumas in Southern California that share a strain. It is thought to affect 1.5 to 3.5% of domestic cats in the United States. It is typically not fatal, but many cats are carriers. FIV is primarily transmitted through deep bite wounds. A FIV-positive cat will bite another and the positive saliva will enter the tissue. Because this is the main transmission mechanism, risk factors include the male sex because males typically are more aggressive, adulthood, and outdoor access. Kittens born to FIV-positive mothers often test a false positive due to the antibodies present in the mother's milk. However, after about 6 months, the kittens will test negative. The virus accesses the host cells through an interaction of the envelope glycoproteins, and targets the cell surface receptors, commonly CD134. Once it has attached to the transmitter, the viral RNA is transferred into the cytoplasm. Once the viral RNA has entered the cell cytoplasm, it is incorporated into the DNA with non-homologous recombination. This can lay dormant for over a year, or it can immediately trigger lysis of the cell, otherwise known as a cell death, which will then release its prodigy. The transmitter CD134 is typically found on T cells, which are critical in the immune system. If the T cells drop significantly, the cats become more susceptible to opportunistic diseases, and this can progress to feline-acquired immune deficiency syndrome, otherwise known as FAIDS, which is commonly known as AIDS in humans. FIV targets multiple cell types besides T cells, including CD4 positive, CD8 positive, T lymphocytes, B lymphocytes, and macrophages. However, T cells are its, quote, favorite cells. There are many common symptoms that come with FIV, including disheveled coat, fever, poor appetite and weight loss, enlarged lymph nodes, abnormal or inflamed eyes, other known as conjunctivitis, anemia, diarrhea, inflammation of the gums, other known as gingivitis, dental disease, inflammation of the mouth, other known as stromitis, wounds that don't heal, sneezing, discharging from eyes or the nose, frequent urination, and behavioral changes. My research question is, are certain strains of FIV more virulent, and do they vary by species? And what this means of varying by species is kind of asking if the clade that they are in, and that's going to affect that species, is it more virulent? Or also it could even be in the scenario that the host has adaptations that make them more resistant to that strain. Why care? Well, nearly all cat species are vulnerable to extinction. This makes their populations much more sensitive to stressors like changes in the environment or deadly diseases that could be affecting them. This is why it can be really important to understand not only how the disease affects the populations, but if there are certain strands that are more deadly and that should be maybe focused on containing rather than going through the population and potentially wiping out the species. Another reason why FIV is really important to research is that it is very similar to HIV. So understanding FIV could lead to a better understanding of how to treat HIV or AIDS, for that matter, in respect to therapy, vaccination, and pathogenesis. How would this be done? Well, blood and tissue samples would need to be taken from both domestic cats and lions, as those are my two target species for this study. It would be much easier to gather the samples from the cats because they are likely to be less aggressive. However, with the lions, it would be necessary to tranquilize them in order to take the samples. In both cases, the lion population and the cats that have samples taken would need to be monitored for the rest of their lives because FIV can take years to manifest, and any clinical symptoms that they exhibit would need to be noted. If the cats were adopted from the animal shelter, we would need to get permission from the owners to continue to monitor the cats and ask that the owners also regulate them for any changes in their behavior or signs of their clinical symptoms. The blood samples would be examined under a microscope to ID if they are in fact FIV positive, and if they are, what strain and clade they belong to. Additionally, the tissue samples that are gathered will be cultured in a petri dish and infected with different strains respective to that clade. So we would have some for the FIV for the Felis castus or domestic cats, and then for the lions, Panthera leo, and we will monitor to see the mortality in the cells in a lab setting. Most FIV studies have been done on either domestic cats or lions because they are much easier to monitor long term, which is necessary for FIV because it can lay dormant for so long. ApoBEC3 proteins, or apolyproprotein B mRNA editing catalytic polypeptide 3 proteins, are important protein inhibitors of retroviruses and retrotransporters. This protein is very important for host species to fight against retroviruses, and scientists link its emergence as an evolutionary arms race between the host and viruses. In cats, they have one A3H gene and three similar A3C genes, which were probably produced after consecutive gene duplication. In a study with FIV positive cats, they found in a house where there was two or less, most of the cats did not display clinical signs of the illness. However, in households where the cats were kept in an environment with 60 or more other cats with unrestricted access to each other, 63% of the cats lost a median of 51.3% of their body weight. Most commonly, lymphoata was the common cause of mortality in these cats. Overall, cats did not have a high mortality rate. However, it appears that they are more vulnerable depending on the environment that they are in. The FIV-PLE subtype is commonly found in lions, and it has distinct phylogeographic distributions, which suggests that it has been in the lion population for a long time. Scientists think that it probably first began in the late Pleistocene expansion of lions. It has a very high prevalence, and this is probably because of the highly social nature of lions, so they are more likely to be interacting with each other and exchanging those deep bite wounds and transferring the disease between each other. In many populations, there will even have 100% seropositive adult individuals, while others they may remain unaffected. This all or nothing distribution of FIV makes appropriate comparisons between infected or uninfected lions a challenge at times. Differences in FIV-PLE effects can also be influenced by environmental factors such as infectious agents, prey abundance, and water availability. Little evidence has been found of the decreased lifespan of FIV-PLE in endemic populations. However, a few clinical studies revealed that individuals of these same species may demonstrate FIV-associated immune depletion and in some cases AIDS-like complications and death, which suggests that this advancement of FIV doesn't just occur in cats, which was not positive beforehand. In Felis catus, or domestic cats, they found that the strain C was more virulent than the strain A. In kittens, the strain C had quite high mortalities, and while in adults there were fewer mortalities, they did have 100 times more mean levels of viral RNA compared to the strain A. After 20 weeks post-infection, strain C still had more proviral DNA and tissues and their lesions were more severe. This difference between adults and kittens suggests that there is a buildup in age resistance as they mature. Lions are commonly infected with FIV-PLE with 6 different strains, and in the graph you see here, there are all the different strains of FIV. Scientists hypothesize that lions were infected 3 separate times with FIV-PLE. They examined a lion population in the southeast Serengeti National Park. They found that the frequency of strains depended largely on ages, and in the population, 12% were infected with subtype A, 57% were infected with subtype C, and 69% were infected with type B. In cubs of 1 year, in subtype B, they were infected 67%, and that increased when they went from ages 3 to 4 to 80%. For subtype C, it was quite rare in yearlings, only about 17%, but it increased by the age of 2 to 69%. For subtype A, it remained low throughout their lifespan. Females showed a longer lifespan with subtype B than A or C, suggesting that both of these types are more virulent, but the trends weren't found as much in males. Also, in a 1993 to 1994 study, they found that the lion populations declined by 39%. Originally, they thought it was due to FIV, but is now understood to be due to canine distemper virus, or CDV. This CDV exposure outbreak killed 67% of lions, but only 57% of the higher risk subset survived. They do know that it is because of CDV. However, they think that FIV could have potentially played a role with compromising the immune systems of the lions. Additionally, subtype C had a higher risk or higher rate of mortality than what was expected. Because FIV has been in the feline genome for so long, the cats have largely adapted to the virus, so it is not as deadly as it may have originally been when it first entered the population. Because of this, I would expect to see similar rates of clinical observations of the virus as well as any mortalities compared to the lions or cats. However, I would keep in mind that the environment could be influential to see if we do have any mortalities. So if the lions have a particularly bad drought or the cats are kept in homes with a quite large number of other felines, I might see more observations of clinical symptoms or mortalities. I predict that in domestic cats we will see a greater mortality with subtype C based on the data that I looked at in literature. That was the more virulent type compared to A. And in lions, I predicted that subtype C would be more deadly. Both A and C were found to be more deadly than B, despite B being more prevalent in the population, and C was more commonplace than A. However, I think that it could be potentially reversed if canine distemper becomes more commonplace in the population, because any disadvantage or weakness in the immune system could lead to fertility in the lion, and B does occur from a young age, while C occurs later in life. Here are my sources for my research. I hope you enjoyed and have a good summer.